So, yes, thank you very much to the organizers for the invitation. It has been a great conference so far, and I adapted my talk a bit to what I felt where the meeting was going, so to have a bit, it's fine, a focus on mechanistic modeling and how it can help us to understand how signaling pathways work, because after all, in spite of all the great successes of the increasing availability of big data, in the end, all of this work is really based on mechanistic insights that have been gathered over decades. So I want to start with a very simple example that many of you may know. So a G-protein-coupled receptor pathway. So you have the ligand that binds to the receptor, then activates the receptor that recruits G-proteins, and then the G-proteins get activated and release G-beta gamma. And one of the nice things that computational models allow us to do is to ask what if. So what if we interfere somehow with this pathway? For example, what if we interfere with the recruitment of the G-proteins to the receptor? Or what if we interfere with the recombination of the G-proteins after G-alpha has dropped back to the GDP state? And we can do this by introducing dedicated inhibitors into this system. And the software I will discuss, so our approach, allows you to do this in a very elegant manner. You just specify the interaction properties of these inhibitors, and then the approach will automatically adapt the computational representation, will allow you to perform simulations of this pathway. So we go from this kind of network to a slightly modified one, and now we can ask, how does the addition of these components shape the behavior of this network? So without these inhibitors, just the native G-protein-coupled receptor network, you provide a stimulus and you can look downstream at the production or release of free G-beta gamma. But now we have these inhibitors and we add them and we perform simulated variations of these concentrations of inhibitors. In this case, we draw the distribution of these concentrations from a log uniform distribution over 2 orders of magnitude. And now, instead of a single curve, we obtain a whole variety of different outcomes. In this case, 1,000 different time courses corresponding to the different doses of these inhibitor concentrations. And obviously, if you just look at these 1,000 curves, it doesn't seem to be very useful. But now imagine that for some reason, you want to push the system here. So you want to have a strong response that lasts. What our approach allows you to do is map back this behavior of your pathway, so a rapid, strong response that stays on for a long time, to the parameters in your system. In this case, the concentrations of the inhibitors. Yeah? And I think you can imagine where you could apply this to more complicated pathways that you want to push in a particular direction. And I will now apply this to modeling the crosstalk between IL-4 and IL-7 cytokine signaling, a bit more interesting biological application. So IL-4 and IL-7 are members of a family of cytokines that require, in addition to the private receptor chain that binds the cytokines, the common gamma chain. So as soon as they have ligated their specific cytokine, they recruit the common gamma chain, and then the associated kinases become activated and activate downstream transcription factors. In the case of IL-4, predominantly STAT6. In the case of IL-7, predominantly STAT5. And so we asked, given that the common gamma chain is used by all of the signaling pathways, is there a situation where the common gamma chain could become limiting? Yeah, so we quantified the expression level of these different chains on the surface of naive CD4 T cells. And what we found is that indeed the common gamma chain is outnumbered roughly 5-1 by all the other chains. And all of them need the common gamma chain to signal. Okay? So we focused on IL-4 and IL-7 signaling because they have quite different biological meanings for the cells that receive these signals. And what we did was we stimulated naive CD4 T cells first with IL-7 alone, and then we added IL-4 and compared this to the outcome you would get when you give only IL-4. And indeed, what we found was that the STAT6 response downstream of IL-4 was shifted, was suppressed by the pretreatment with IL-7. And this was an effect that was dose-dependent. As you increase the concentration of IL-7, the suppression of the IL-4 pathway and also of, similarly, the IL-21 pathway was increasing. Now, if you look at the expression levels of the private receptor chains, one explanation that might immediately jump to your mind is that, well, you know, if you ligate the IL-7 receptor chain, it just takes away all the common gamma chains and there will be nothing left for the IL-4 chain. Knowing the KD of the interactions between the cytokines and these chains, we can translate this into the number of bound IL-7 receptors, so these doses. And what we find is, surprisingly, that roughly 10 bound IL-7 receptors can exert a very strong suppression on the IL-4 system, which is quite surprising. Yeah? If you take away 10 of these roughly 600 common gamma chains, how can it exert such a strong effect? So we were left with a quantitative riddle that we tried to solve with a quantitative computational model. So we have IL-7, the IL-7 receptor, the associated kinases, the common gamma chain. We have STAT5 in this case for IL-7. We have phosphatases and some other negative inhibitors. So, and now if you count all the different states you have to model, and this is a very innocent looking system, yeah? So you have 2 states for the receptor, then you have different states for the STAT side, and so on. And you end up with 96 states here for the IL-7 system, 4 for common gamma, and overall, you have 384 states in this system, which is really a simple system. Yeah? So if you want to simulate this with differential equations, you need 384 coupled differential equations. Yeah? So the typical approach of doing this by hand seems a bit tedious. That's why we developed a new approach where you can use an iconographic language. Here, for example, the IL-7 receptor with the extracellular domain binding to IL-7 and different other binding sites. And importantly, we have these tags that encode different states of the molecules. So this, for example, is an activated kinase, and this is the inactive kinase. And we can use this iconographic language to encode interactions and reaction rules. Here, for example, we can say, if we have this heterodimer of the IL-7 private chain and common gamma, then we have the activation of the associated kinases. And based on the user-specified pairwise interactions, our approach will then automatically build the computational model. So, and in addition to that, you get a nice network view that you can query. So the good thing here is if you build such a model and you want to share it with a collaborator, it's a computational model. But to share it with someone else, you're not sending them a bunch of differential equations, but you're sending them this, and they can look into every detail of your model. There are no hidden assumptions, nothing. Yeah? So they can really look into what is your model actually assuming. And so we implemented the textbook wisdom that without the cytokine, there is a low affinity binding between the common gamma chain and the private chains. And then with the cytokine binding, these interactions become stronger and the complex is more stable. So we went from these molecular mechanisms to a computational model, and we ended up with roughly 40 parameters, which seems to be standard for this conference. Now, 40 parameters, if you have ever talked to a mathematical modeler, they will tell you 40 parameters, you can fit anything, yeah? The famous John von Neumann quote, give me 3 parameters, I fit an elephant, give me 5. I make it wiggle its trunk or his trunk, whatever. Well, that's what we did. We varied all the parameters, and these were the levels of suppression we could see. Yeah? You see, if we add a lot of IL-7, the phosphostat-6 response goes down, and the suppression gets weaker if we add less IL-7. However, these were the experimental data. Yeah? So even though we had a very nice, so these data actually represented or reproduced the experimental findings for IL-4 alone, but they completely failed for the IL-7 dose dependence. So we came up with an alternative model. So second hypothesis, maybe downstream of IL-7 you activate a phosphatase, and this phosphatase interferes with the activation of STAT6. Well, you can test this experimentally. What we did was we looked at the decay of STAT phosphorylation after different doses of cytokines and after inhibition of the kinases. So this gives you a readout of the phosphatase activity. And we found that, yes, the phosphatase activity may go up a bit after cytokine stimulation, but in our simulations, we could look at how would the phosphatase activity have to go up to reproduce the data. So from these circles to the triangles, and these are the experimental findings. So there is no way phosphatase activity regulated by IL-7 can explain the experimental data. So we did something that many of you told us here during this meeting. We released a couple of the parameters in our model. Specifically, we said, okay, maybe there is a medium affinity interaction even without the cytokine. Yeah? And now, suddenly, we were able to reproduce very nicely the experimentally observed suppression of IL-7 on IL-4, and we could make nice predictions about the hierarchy of the affinities between the private chains and the common gamma chain. And these findings were nicely confirmed experimentally. So, for example, the dependence of the crosstalk on the level of IL-7 receptors, and then we performed breadth measurements measuring how well the IL-4 private chain can interact with common gamma, depending on whether you add the IL-7 private chain or whether you add, in addition, the cytokine IL-7. And we can look at it, because we are simulating this, we can look at this in a dynamic way. So during the equilibration, we see how the IL-4 receptor binds to the cytokine, how this is basically reduced through the accumulation of the common gamma chain on the IL-7 private chain. And then when you add the cytokine, if you compare without IL-7 and with IL-7, you exactly get this reduction that corresponds to the dose. Note this is a very small dose of IL-7 that we found experimentally. So the take-home messages, so even though there is a strong pre-association between the common gamma chain and the private chains, the system still responds to cytokine stimulation, which is kind of surprising, but which we could explain through the quantitative model. And second, if you look at it, that if you think about it, that there are only 500, 600 common gamma chains and 5 times more private chains, you might think this is some kind of a bug of the system. Yeah, but it might be a feature because it allows the immune system, it allows these cells to tune their cytokine responsiveness. And the immunological interpretation is that depending on the history of exposure, IL-7 to IL-4, the cells show different sensitivities. And in particular, people have always assumed that when cells after activation proliferate, they downregulate the private IL-7 chain. This is out of altruism because they want to leave IL-7 for other cells. But another explanation may be that they have to downregulate the IL-7 chain in order to become responsive to IL-4. Yeah. Just one quick outlook beyond single cells, because this was also a theme on this meeting. So we can simulate these signaling pathways, not just in a well-stirred system, but we can simulate them actually in cells with arbitrary morphologies. And we are working on combining image analysis where we extract extracellular structures and then populate these extracellular structures with cells that carry each cell within them signaling pathways that make them respond to the extracellular environment. So here you see a simulation of cells that have 2 kinds of signaling pathways. One that allows them to migrate in this direction, so a chemokine receptor signaling pathway, and an adhesion pathway that allows them to migrate on the fibers. And you can imagine how you can use this technique, for example, In this case, we're looking at collagen fibers in the ear of a mouse that were collected with second harmonics images. But these structures obviously could also be blood vessels or anything, and we could simulate really cell-cell interactions and cell-environment interactions based on individual pathways acting in cells. And I would like to encourage you to try the software. It's free, obviously. You can download it from our website. If you want to register, that's great. Then you get updates. You don't have to. And for those of you who consider themselves beyond iconographic modeling, for the real hardcore people, you can use Python to access most of the functionality. So there you go. And I want to acknowledge some of the people here. Pauline Gonor, she was, together with Rajat Varma, very strongly involved in this IL-4, IL-7 story. Bastian Angermann and Fengkai Chang were the main programmers behind the modeling approach. And I want to acknowledge that the work was supported by the research program of the NIID, and I'm happy to take questions.
We have time for one question, if there are any. Right here, Marcus?
Thanks, Martin. This is great. One question I had was, when you looked at the changes in the parameters, you found one thing, but I presume you looked at multiple things.
Yeah.
So what was that process like, and were there other possible candidates?
Yeah, so we, as I mentioned, one other candidate, the phosphatase activation. So we looked actually at different stages of phosphatase activities at the level of STATs, at the level of the JAK kinases. And the pathway is relatively small. You've seen all the ingredients. So the number of different possibilities is limited. And the fact that in the end, we obtained this very nice correspondence between the model and the experimental data, and also with regard to this prediction, regarding the hierarchy of the binding affinities, was quite encouraging.
Yeah, that's impressive. Thanks.
Thank you so much.